very excited that we have representatives from the Science Museum of Minnesota here with us today. So Adam Heathcote is the director of the Department of Water and Climate Change. He has been working on northern waters since 2015 and previously studied lakes and ecosystems in Quebec and Greenland. He has a PhD from Iowa State University, where he studied some of the most polluted lakes in Northern America and learned firsthand the importance of protecting our few remaining wild and scenic places. And we are also hearing from Allison Rempel Brown, who is the CEO and president of the Science Museum of Minnesota. He joined the Science Museum of Minnesota in 2016 with a firm commitment to the mission and advancing and centering equity within the mission. Before the Science Museum of Minnesota, Allison served as the chief of staff at the California Academy of Sciences. She's a graduate from Pomona College and the University of Chicago Booth School of Business. And if you do a little bit of looking into it, you know that she was honored as a person to watch by the Minnesota Business Journal, MSP Business Journal, and the Business Exec of the Year by the Minnesota Business Professions America. That's pretty neat. We're really lucky to have these folks with us here today. Yes,
I'm looking at how this wonderful opportunity came to us. How did it come about?
Well, the wonderful network that UV Minnesota has and the wonderful network that Tuesday Group has. So Allison Hellman? Jessica Hellman. Oh, gosh, it's Allison. Jessica Hellman is a new member of the board of the Science Museum of Minnesota. And you may remember her name as a former Tuesday Group speaker and an active member of our community. So we thank Jessica for her connecting us to more fabulous folks. Awesome. Welcome. Come on up.
Thank you, Lacey. I was at the museum in our lobby the other day, and I hope some of you have been to the museum, and if not, I welcome you to come. And I was in our large lobby, and I ran into a grandfather who was there with his grandson. And the grandfather met me and said, I am so excited to be here with my grandson, because I used to go to the old location with my daughter. My wife and I would take her. And to come here with my grandson reminds me of that time. And now when I come with my grandson, we do many of the things that I used to do with my daughter. And there are a lot of new things. But when I come with my grandson, the first thing we do is we come to this big lobby and we have to go wave hello to the T-Rex. And then we run across this big lobby and go to the musical stairs that you had at the old location. And we have to run up those steps and then look out the beautiful view of the Mississippi River and see what's happening. Because depending on the season, there's always something new in the Mississippi River. And then I had to get myself ready because my grandson's going to go running down those stairs into the exhibit area. And depending on my grandson's mood, I don't know where he's going to go. We may go down the next level and go visit the mummy and say hello to the mummy as well. But what we always do, we always go down to the bottom level to that new exhibit you have. And I can't remember the name of these. But he can race a T-Rex. And he never, ever wins when he races a T-Rex. Now, a little aside here. This is for me. You can also race TC Bear. I've never beaten TC Bear, so I think I got to work on my exercises. But back to the grandfather. The grandfather said to me, thank you so much for making this experience possible. Because when we walk around that bottom floor, we can try all sorts of different experiments. And this sparked my grandson's curiosity about all sorts of science things, about what he can learn and potential that he can follow that also inspired my daughter as well. And it was just so moving for me. And I've heard these stories frequently from visitors who've been to me. We get visitors to the museum from all over the state of Minnesota, and frankly, from all over the United States, from all over the world. And that's one of our superpowers, is that shared experience with family and friends. What people coming to the museum may not realize is we have another superpower. And that superpower is science and education centering equity. Because what visitors may not realize is that the Science Museum is more than a museum. We also provide educational resources in all 87 counties of the state. We provide these for teachers, for students, throughout the state, through all of our programs, as well as professional training for teachers. And we did this during the time of the pandemic. I don't know if any of you heard this report that came out recently, that during the pandemic, students lost 10 years, a decade worth of training, a decade worth of learning during this time. We're also a STEM pipeline builder. Now, STEM is science, technology, engineering, and math. We have a particular focus to help those who've historically not seen themselves going into STEM careers. That would be women, girls, Black, Indigenous, and people of color. And we have a shortage of talent in STEM careers already. In the next 10 years, STEM careers are going to grow 3 times faster than all other careers combined. 3 times faster. We need to find ways to get more people see themselves going into these careers. At the Science Museum, whether it's in our location in St. Paul, online, or throughout the region, we also are a place where we can reflect and have conversations about some of those important topics that we need to talk about today. Climate change, systems of oppression, and how we can all be part of ways to create solutions and develop a society that we all strive to be in the United States. And we also do original scientific research at the museum around these important topics and how we can help more people go into STEM careers and implement the solution we already have today. Now, the pandemic isn't over, but that crisis really led us to think about what do we need to be as an institution. And we saw that science was one of the superstars during the pandemic. As this crisis, this health crisis, really drove us to think about where we move and where we go forward, and how we recover from this pandemic. How do we change our systems? How do we change our thinking? How do we change the way that we move forward? Can we hear in the back? Is there an echo? A little closer. Okay. Raise your hand if it gets too soft. Won't hurt my feelings. So as we did that, we really leaned into in developing something. Thank you.
Does it work?
This is what happened when I pushed the wrong button.
Yeah. You're not the only one.
Into developing our strategic plan, looking forward to 2030. We will build bold science experiences to spark curiosity around science. to spark imagination. We will also develop the STEM pipeline needed to open up those STEM careers. And we will lead and partner around science, around matters that are so crucial for our society. Whether it be climate change, conservation, sustainability, what does it mean to be human? And how do we identify around being human of systems of energy, health, livelihood, all of the things that we need to have conversations about together. This transformative and important work is really leading us to reflect and consider, what is a museum? Who is a museum for? How do we meet and consider the needs of our changing audiences and communities? And how do we answer a phone?
Sorry.
That's why. And we do this work because of people like you who support us. So really, I want to thank you all. I had such great conversations talking with you. And this is really continuing the work that many of you have done throughout your careers and the thoughts that you've had as you move forward in your careers and your leadership. It works. We want to partner with you. We want to be your partners. And we are passionate about making certain you have a future that can use science moving forward, and we stay engaged with you. I enjoyed my conversations talking with you and learning about your passion, what you care about, and how you will be moving forward with your lives. And one of the things we are doing, we're bringing here to you, which is an example of our bold science, which actually is a continuation of some of the work that you've spoken to me about. And I introduce the Science Museum's Director of Water and Climate Change, Dr. Adam Heathcote.
That one. Thanks, Allison, for that introduction. Thanks to all for your attention. I'm going to try not to step in front of this table, but I am known to move around while I'm speaking, so feel free to stop me if I get too far. Okay, so as Allison said, I'm Dr. Adam Heathcote. I'm the Director of the Department of Water and Climate Change at the Science Museum in Minnesota. And I'm going to talk to you about some work that we're doing. I'm actually just starting. You are all lucky. This is the first time any of this stuff has been presented to an audience. I'm going to talk about two brand new projects, one of which just started just months ago, both kind of centering around North and Northeast Minnesota, but both dealing with the same kind of issue, and that is algae blooms in places we don't want them, namely our streams and lakes. This picture here was just taken actually last month. This was up on East and West Twin Lake in the Gunflint Trail, just north of Grand Marais. So as Allison said, one of our superpowers and one of our best kept secrets, although we're trying to share it more broadly now, is that the Science Museum is more than just a place to go visit, although that is something that is very valuable to us and to all of our visitors. We're also a place that does science. So we have a scientific staff in a number of different disciplines, including anthropology, paleontology, biology, and the department I work in, water and climate change, is made up of primarily ecologists and biologists who work with freshwater systems. The reason the museum formed this department was to increase the impact of our environmental and science advocacy efforts, to have a unified approach of these two intertwined things, universal need for clean water and clean air. So we saw a picture of the Science Museum, the main building down in St. Paul right on the Mississippi River. And we also have a building on one of the other major rivers that runs through the Twin Cities, the St. Croix River, the National Scenic Riverway. It's one of the first wild and scenic rivers designated by the National Park Service. And the St. Croix Watershed Research Station, which you see pictured here, is right there on its banks. We're a team of about 15 scientists. Most of us, as I mentioned, working on issues with water quality and intersection between what happens on the land and how that affects the water. So another one of our superpowers is that we are a great nexus for a lot of different groups who are interested in water quality and all the different benefits they have, whether it just be the intrinsic beauty of water, whether it be the economic or environmental benefits of water. We can work with all of these groups because the Science Museum is uniquely trusted as a source of scientific information and expertise. We work with state, federal agencies. We work with indigenous resource managers. We work with local conservation districts, county conservation districts. We work with industry. All of these people working together for the same mission in Minnesota, which is to protect our water quality. So as I mentioned, I'm going to talk about very briefly about two studies. And I can only talk about them briefly because they literally just started. The first one is on something called Didymo or rock snot. So show of hands, who here has heard of Didymo or rock snot? One person. Are you brave enough to tell me what you've heard about it?
Well, it is a really gross growth that infects some western trout streams. That's
Yes.
why
Did you know it's in Minnesota?
I did not know it.
Okay, well, I'm going to tell you a little bit about it, the history of it. So yes, you're correct. It is an algae. I've talked to a couple of people about this beautiful group of algae called diatoms. One of the reasons they're beautiful is because unlike other groups of algae which have carbon-based cell walls, diatoms actually have silica or glass-based cell walls. This is actually a micrograph of the algae in question here. Didymosphenia geminata is the scientific Latin name. We can call it Didymo for short because it's less of a mouthful. Here growing, you can see it's very beautiful. It actually grows naturally in Lake Superior. It's been seen there at least to the 60s, probably hundreds of years before, but that was the first time anyone actually took a sample and measured it. But unfortunately, or perhaps fortunately, up until about 2018, it was never seen in any of the North Shore streams. So as you mentioned, it makes this gunky, slimy stuff. That's why it gets this other colloquial term, rock snot. But it only does that in streams because in lakes, two of those lakes where it's most famous for living are Lake Superior and Lake Baikal, two of the biggest lakes in the world, have huge splash zones, and then basically the waves beat it back on the shore. So this is a natural growth. Some of you might recognize where this picture is taken near Artist Point in Grand Marais. This is what Didymo looks like growing in Lake Superior. You'll see here is another micrograph of it. And as you mentioned, it has been invading streams all over the world, actually. So you can see the map here of the world are places where Didymo has invaded streams where it has not been before. And so you can see there's a big cluster in the western United States. People who are worried about trout, specifically like endemic trout species in western United States, are really concerned because what it does when it gets into a stream, without that constant wave action, it just basically fills it full of this knotty mess. And that knot, if you will, I'm trying to say it too many times, is this mucilaginous stalk that the diatom itself grows on. But it's not actually the diatom cell. As I said, that's made of glass. It's this polysaccharide mucousy stalk that it grows to attach itself to the rock. And it actually becomes so great at producing this, it can basically choke a stream completely from bank to bank. So here's the bad news, just the Didymo goes bad. So as I mentioned, Didymo has been happily been living in Lake Superior for 60, 70 years, probably longer, without ever coming into any of the streams along the North Shore or anywhere else in Minnesota that we know about. However, in 2018, it was found by a DNR officer in the Poplar River, which is just near Lewiston, Minnesota, if you're not familiar with. This was the first ever documented case of Didymo in a stream. And it was actually quite a ways away from Lake Superior itself. It was actually in Superior National Golf Course. Well, not like it was directly at the mouth of the river. It had moved up quite a bit, and so this baffled us. For one, how did it get there? Why the Poplar River? We had all kinds of hypotheses. Well, it's probably an isolated thing. Maybe it has something to do with like snowmaking at Lutsen Mountain because they piped Lake Superior water up to make snow, and so maybe there was, somehow the diatom got there, and it's not a big deal. But we thought it was worth looking into because again, this had never happened before in any Minnesota stream. So we were funded by a number of different agencies, including the DNR, the Environmental Natural Trust Fund, Minnesota Sea Grants, to kind of look at this in a systematic way all along the North Shore of Lake Superior. And that's demonstrated here. This is turning on the science. So we kind of went crazy on this, if I'm being honest. So we have scuba divers out in Lake Superior doing depth transects, looking at the natural populations, because we're interested also if it starts to grow deeper into the lake, and by deeper I mean like 6 to 30 feet deep, it could start actually impacting some of the really valuable cold water fisheries. So that's where all of the hatching grounds for things like Heathcote and lake trout are in shallow North Shore Lake Superior. So we had scuba divers out looking for it. We had teams all in the streams, all up and down the lake shore. You can see this map here, the North Shore Lake Superior. The green dots are paired lake and stream sites that we monitored last year. The red lines are transects where we actually went out into the lake with scuba divers looking for it. And here's what we found. The 2021 Didymos shock is what we're calling this. So we found it in, let's see, 1, 2, 3, 4, 5, 6, 7, 8 rivers along the North Shore. So not only in the Poplar, but 7 additional rivers. And actually the Poplar wasn't even close to the work one. Devil Track River, if you're familiar with that one, just north of Grand Marais, which is a very valuable coastal brook trout nursery, was actually the worst one. It was actually choked from bank to bank with this rock snot thing that we were worried of. And it went for miles up the stream. So basically the first time we looked for it, we found it everywhere. So now we had to take a step back. We were hoping this was kind of an isolated, some kind of maybe engineering problem that could be fixed, because we thought maybe we were just accidentally moving Lake Superior Didymos into the stream, but that's clearly not the case, because where we happened to find it in the Poplar River wasn't even the place where it was the most fun. We came back again this year. Now we found it in 1 additional river, 2 islands, as well as a huge growth of it in the Kimball River. So it was present there last year, but this year it's kind of gone crazy. There are several different hypotheses about why this might be happening. One thing that we know Didymos likes is clean, cold water. So most of you, when you think of algae blooms, you probably think of, oh, that's a southern Minnesota problem. That's a, agricultural pollution, or an urban Minnesota problem. That's stormwater. But it's not the case with this algae, because it's very clever in that it's one of the only algae that blooms in the most pristine environments. I mentioned the 2 of the places famous for being native to is Lake Baikal and Lake Superior, 2 of the lowest nutrient systems in the world. And what it does is by making those kind of crazy mucilaginous growths, it actually has this symbiotic group of bacteria that live with it that strip organic and mineral phosphorus out of the water that other algae can't. It is not available in other groups of algae. So when we're out there measuring, water concentrations of phosphorus, which is 0, 0, 0, 0. That's not the phosphorus that this species is going after. It's going after those types that we don't usually consider to be bioavailable. So it's an interesting problem. It's kind of turned the algae bloom problem on its head. And so the question here is, what do we do about this? Of course, our usual tools in our toolbox are, okay, reduce nutrient loading, increase perennial vegetation on the landscape. They're not really the issue here. So this is more of a biological and evolutionary problem. Why is this group? Why did Didymos all of a sudden decide to move from Lake Superior, where it had been for at least 70 years, into the streams of the North Shore? And that's an open-ended question. We don't have an answer to that. What we're doing is we're actually for one, we're going to get the first genotype. So we're going to sequence the entire genotype, this species. And then we're pulling samples from all over the world to figure out Did something change in the DNA of this species to allow it to do this? So is this a microevolutionary problem? Is it a new strain that invaded and is not behaving as well as the native Lake Superior strain? Or has it actually evolved in place to move into these areas? Or the more worrisome problem is this an environmental change problem? So is this the streams are actually changing and becoming more easily invaded by this species that was in Lake Superior? So are they becoming warmer? Are we having more frequent droughts? So I mentioned that it likes very clean water. Well, last year, those of you who live in northeastern Minnesota know was a terrible drought year. Almost all of these rivers were at base flow. And so this is the perfect condition for this species to live because it doesn't have that sheer force of the rivers moving to knock it off the rocks. The water is very clean and clear because it's basically just groundwater seeping out from the rocks. And so this is something we're going to continue to watch. Again, we didn't see as much of it this year. Of course, we had a lot more flow. A lot of you probably remember the springs that you were crazy on the shore. So this is something that is a work in progress. This is actually, we're going to be out here for at least 3 more years sampling these same set of sites, hopefully getting a wide range of environmental conditions to figure out when it flourishes and when it doesn't, so we can get an idea of what the heck we can do about this. So here's what it looks like. These are all taken from Minnesota. I'm putting these up here because this is going to be a give and take presentation because I want you all to walk away knowing what this looks like. So that if you see it, you can tell me about it. Because as with working in any area like northern Minnesota, one of the biggest obstacles is the effort it takes to get out to some of these lakes and streams. And that's where you all come in because you're all out there all the time. A lot of these projects got started because some citizen scientist said, hey, that doesn't look right. I've lived here for 30 years. I've never seen this before. What's going on? I sent a sample. So that's something I want to encourage. I want you all to take a good look at this. And if you see it, because it's not just a Lake Superior stream problem. This has started to show up in some other streams, not in Minnesota yet, but in Michigan and Wisconsin that are not connected at all to Lake Superior. And so how those populations got there, we don't know yet. They've also sent us samples from those areas, and we're going to sequence their DNA as well to see where they're most closely related to. So I mentioned it's kind of slimy and snotty. This is actually from the Devil Track River. This is a rock, if you can believe it. You just picked up, it's just covered in the video here. This is kind of like when it just starts. These tuff are kind of the indicative a sign of it. So these are actually these little guys they're sitting on these little stalks waving away, and this is before it's taken over. This is kind of an intermediate growth, and this is, don't worry about it, this is good. This is what our stream should look like. You know, you want terrifying because that's where all the bugs live, and fishing. So I don't want you to go walk away thinking that if you see algae on a rock in the stream, it's bad. That's not the case. It's this, this coverage of flying that we're worried about. So what have we done so far? Again, we've only been at it for a year, and what we found completely changed what we were thinking we were going to find. So we've been doing lots of outreach. Perhaps you've heard some of it. We've had several stories on WTIP, North Shore Radio. This is an interview we did with Joe Friedrichs right in the Devil Track River. This was during the bloom last year. We raised awareness about it. What I'm doing here today, talking to you all about it. So we want people to know where it is so that they can take action. So one of the best actions you can take, especially if you're a fly fisher person, is that using felt sole waders has been pointed at as the primary way that this has spread all throughout the world. So there's a population that invaded New Zealand that came from Alaska, and they figured out it came from someone who was fly fishing in Alaska, went to New Zealand wearing felt soled waders. Things can live forever in the felt in the soles of those waders. So you can use rubber soled waders. That's the way. I know it's more slippery, but they are like the perfect microbiome for transporting microorganisms. So avoid that if you can. And trying to get people on board through voluntary action, so getting outfitters on board, getting outdoor store on board to stop using these kind of felt soled waders, because where they do have the advantage of additional traction, if you can't fish in the river because it's so full of this rock snot, then what's the point? The other thing we've been doing is education. So we're up every month all along the North Shore from Duluth to Grand Portage, and we always take time to stop anyone who wants to ask us what we're doing. whether it be a group of kids swimming on the beach, whether it be training the next generation of scientists. We've had interns every year working with us, whether it be working with agency personnel, teaching them how to identify this organism, all of the above. One of the other kind of exciting end results from this is Allison mentioned that we reach all 87 counties with our STEM educators. As part of the Sea Grant project, we're actually going to develop curriculum that we can give both in person and virtually to northern Minnesota classrooms, where we incorporate this into the curriculum. So giving them some sense of place by saying, hey, this is what's going on in your area. This is why it's a problem. This is what you can do. This is how you can report it. This is what you can do about it. Teaching, and this, this is how it affects everything from, the little microscopic bugs to the trout. Okay, that was number one. Number two, this one's shorter. Don't worry, because this one really just started. This one, our first field season was in June of this year, and this one is looking at the other kind of algae bloom I talked about, the kind of the one you may be more familiar with, the blue-green algae bloom, or what we call cyanobacteria. So these are the algae that typically grow when you have too much nutrients in a system. These are the ones that make those really nasty blooms in metro lakes and in southern Minnesota. Something that you would hope you would never see in northern Minnesota. However, something that we've increasingly noticed either through satellite photography. So here we're looking at the smoke flame burnt change off Sawbill Lake in the boundary waters. You may not even be able to see burnt lakes because it's almost the same color as the forest surrounding it. See, flame is a color that you would expect a lake to look like on a satellite photograph there, dark black or very, very dark blue. And what we're seeing is they're starting to turn green, and we don't, this is another one where, unfortunately, I don't have the answer for this one yet, because again, we're just starting. We don't know why it's happening. And it's not just in Minnesota. This picture here, my colleague Mark Edlin, another scientist at the Science Museum, I bet you would not guess where this algae bloom was seen. This algae bloom was seen on a lake in Isle Royale. So this is probably the most pristine area in the country, an island that is entirely a national park, not just a national park, a national wilderness in the middle of Lake Superior. We saw one of these. And so, what is going on here? What is causing it? We've got a couple of different hypotheses. The first being one that you may have already kind of thought about, since I've heard there's already a climate change group here. So one thing we're interested in is looking at how the warming climate affecting algae. How might warming climate affect? Well, there's a couple of reasons. One, cyanobacteria, the blue-green algae that we're talking about here, are microorganisms. They're microorganisms that prefer warmer temperatures. So they do better with less nutrients when the water is warmer. So they can make more biomass per unit phosphorus in a warmer climate. The north temperate zone, the boreal zone, subboreal zone, whatever you want to call northern Minnesota, is one of the fastest warming places in the United States. We can see that already, whether you're looking at ice records. This is from Lake of the Woods. This is the number of ice-free days. So we have 30 more ice-free days on Lake of the Woods now than we did in 1960. And why might that be important beyond just the temperature argument I just said? More ice-free days means more time to grow. That means a longer growing season, a longer photo period. That means more time to get the inertia needed to form these large blooms that we worry about. And the other one that maybe you didn't jump to right away is atmospheric inputs. So we're lucky here in the Superior National Forest and the boundary waters. This is one of the most protected places in the world, but we can't protect what comes in from the atmosphere. And so one of the things we're looking at is how do atmospheric inputs, either natural or human produced, affect some of these lakes. And we did a little bit of a pilot study here. We went out to a bunch of bogs. Some of them in like the Red Lake peatland, where there's nothing around them, like no watershed. Anything that falls into this lake came from the air, basically. And what we did, we took cores, so we took sediment cores, we dated those cores, we measured the amount of mineral input, which must be coming from the air because there's no mineral inputs from the watershed. Over time, we see this ramp up, even in these bogs. And at the same time, we measured the amount of algae in this. I talked to a couple of people about this field called paleolimnology. I'm a limnologist, so I study inland waters. I'm also a paleolimnologist, so I study the history of it. How we do that is we take sediment core from the bottom. The top of that sediment core, the part that's touching the water, that's the most recent, that's today. And as you go back in time, the deeper you get, the older it gets. And we can actually use radioisotopes, so things like carbon-14, to date each individual section. So we could tell you, 3 inches is 10 years ago, 6 inches is 20 years ago, and so on and so forth, all the way down to 200, 500, even 1,000 years past. And from this kind of data, we can kind of see how things changed before we were looking at, which is important because we've only just started looking at a lot. And so in this example, we see dust inputs are closely correlated to increase in algae in these bog systems. So at the same time we're monitoring temperature, we need to also be looking at what's coming in from the air. And those things are not completely independent of each other. One of the big atmospheric inputs that you are probably very familiar with is ash from forest. So what are we doing about it? Well, this is the first of its kind study in Minnesota. It's actually one of the first of its kind studies in all of North America. So there's this network that goes all across North America, US and Canada, called the National Atmospheric Deposition Program. And this was put up in the early 90s in response to acid rain. Well, this has been measuring since that time sulfate, nitrate, other things that were acidifying our boreal systems 30 years ago. Luckily, we've turned that around. That is a success story. But they've kept it going, which is great because we can use that now by adding a little bucket. So when they collect these samples, they're only collecting what comes out in precipitation. So we basically augmented it to also collect what happens just from dry. And that those combined two things is everything that comes out of the atmosphere. They collect this every 2 weeks. So we've set up a network that basically wears off the Superior National Forest in Minnesota. That's the yellow dot on your left. There's one in Voyagers, there's one at Marcel Experimental Forest, there's one in Hoagland. I'm trying to remember what the other one is. Well, you can see it on the map. I don't remember what the state is called. And then we selected 8 lakes. They're all on the east side, but that's more east side of the boundary line. That's more kind of logistical reasoning. We can sample all of those 8 lakes in a week, basically. Each one's quite complicated to get to, as you might imagine. And we kind of set it up as a paired study. So I mentioned we were on East and West Twin. They're kind of a perfect pair. So we have East Twin here, shallow. We know it bloomed when we see it from satellite photos. West Twin here, deep. We haven't seen it bloom in any aerial photos. So it's a perfect pairing because we're looking at what makes a lake sensitive to either climate change or atmospheric inputs, or more sensitive, so that it might start changing quicker. The other thing we're doing, besides monitoring, is we're putting buoys in all of these lakes. So these buoys are measuring every 30 minutes temperature and oxygen from the top to the bottom of all of these lakes for 2 years straight, basically, trying to see what's going on with the actual physical structure. So some of you might be familiar with how lakes separate into layers in the summer. So we think this is something that is very sensitive to a changing climate. So as you become warmer quicker, you erode what's called the thermocline. That's that very important density layer that separates those two things. And so lakes that are shallower are going to be way more sensitive to that than those that are deeper. And so they may be our canaries in the coal mine. Those are the ones that are going to start to change the quickest. Again, these are hypotheses. We just started this in June. So what do we do when we go out? We're out collecting water samples. We're collecting them from the top and the bottom, because as I just mentioned, that might be an important thing. We're collecting sediment cores here. This is a new postdoc who just started in May, Dr. Leanne Sefna, who's leading this project. This is what one of these cores looks like. So we can actually take a lot longer than this, but for a lake, this one is from Finger Lake, just north of Coffee. For a lake of this size and this productivity, about just under 3 feet is enough to go back about 300 or 400 years. So enough for us to cover the time period of interest. I just like this picture because the water she pulled off the bottom was like yellow. And that's nothing wrong with the water, but it's just kind of something interesting that you see in some of these boreal or subboreal lakes, because you get a concentration of the dissolved organic matter at the bottom. And so it's just kind of cool. You have clear water at the top, you pull it up from the bottom, and it looks like lemonade. So it didn't take long for us to find our first toxic algae bloom. As I mentioned, we just started. This was last week, actually. So last week we were out, this was the lake where we just had the picture of the core one. This is kind of what these look like. A lot of times they're on the windswept shores, so that's a good place to look for it if you're interested. And that's just because they kind of just get blown into big colonies. And this is a species of cyanobacteria called Gliotrichia cumulata, which is pictured here. This is a micrograph. That's what it looks like under a microscope, but you can actually see it with the naked eye. It looks like a little fuzzball floating. And this is kind of one of the early warning signs for bloom, because this is a very crafty cyanobacteria because it actually can control its buoyancy. And so it'll move down to the bottom of the lake, scavenge nutrients that are down in those bottom waters. And then it'll do that at night. And then during the day, it will migrate to the top where it can now use that phosphorus it grabs in conjunction with the light from the sun to form biomass. A lot of algae cannot do that, just to be clear. Most algae, they grow in the photic zone, they sink to the bottom, and they die. This one goes up and down, up and down, up and down, whatever suits it. How can you help? I mentioned this is going to be a give and take, so I'm going to ask something of you. So there are a couple ways. So I'm going to start with the Didimo project, the Rockstop project. Science Museum are big supporters of citizen scientists, and how you can actually provide data to us that we use. There are two websites. They're actually linked. It doesn't matter which one you do it, which one you use. We'll get the data either way. The first one is called EdMaps Midwest. The website is there. If anybody wants that information afterwards, if they can't get it off the screen, just let me know. I'm happy to give you the link. This is run by the Minnesota DNR, but both myself and another colleague are actually expert verifiers on here. So if you put an identification of Didimo or Roxanne up to here, we will look at it. We'll get a notification and we'll look at it and we'll see where it was, when it was, what if we can tell it's Didimo or not. And then we can kind of start to map where it might be besides on the North Shore. You can also use iNaturalist, which is even more user-friendly than EdMaps. All you need to use iNaturalist is a cell phone, take a picture, it automatically georeferences it and uploads it. It will even tell you what it thinks it is. And the nice thing is iNaturalist actually routes all of its observations to this EdMap website, but we'll then get them and be able to verify them as well. So if you put data into there, it will be used. The other one you can do is, as I mentioned, kind of the reason that this problem with algae blooms in wilderness lakes has not been looked at before this year is that it is just so hard to get into. We have so many lakes here, and some of them are so hard to get to, especially in this area. But you are all out here all the time. And so you are the people who can send us photos and locations, and it will never bother us if it's not actually an algae bloom. That will make us happy. But if it is an algae bloom, that is a data point for us. That is a lake that next time we're in the area, we might go look at. And so all you have to do is send a photo. I put the research station's email here, scwrs@smm.org. Send us photos. And the best kind of photos are like if you take a photo of the bloom from like stepping back, so what does it look like from the shoreline, kind of the general extent of it, and then a real close-up. Because for a lot of these species, they form these big colonies, and we can actually tell what species they are, even from a macroscopic image. So if like this particular species is called a Phanozomenon blattaqua, you may have seen it before. It forms what look like little floating leaves of grass. And it, if you sent us a photo like that, we'd know what it was right away. So send us photos, email us, use iNaturalist, EdMaps if you see anything that looks remotely snotty in any stream. If any of this was of interest to any of you, you can stay up to date on all of our work in northern Minnesota. We work a lot in northern Minnesota. I have two teams. I basically have a team in Minnesota almost every 2 weeks in northern Minnesota this summer, and we'll next summer as well. So we're all over the North Shore and the boundary waters of Superior National Forest. You can follow us. We have Instagram, Twitter, Facebook. Both the Science Museum and the Research Station have their own. And we post lots of photos. We love to post photos. So you can follow us there. And also, I encourage you, if you see us, here we are out coring Elbow Lake in the Superior National Forest, paddle on up, just like this person did, and ask us what we're up to, because that does look kind of weird. But here we're taking a sediment core. We've got about the 20 feet of rod attached to a coring device that we're driving into the sediment. And this person's like, what are you guys doing here? So, and we love that. So feel free to do that. You see us out there. Pile up and ask us what we're up to. And with that, I think I hope we have time for questions for Allison or Adam.
And we do have a few Zoom questions that I'll relay, but thanks to you both. This has been really wonderful. I have two questions. The first, I noticed in the beginning on your list of partners, you had the National Park Service, but you didn't have the Forest Service. And so much of your work is in the Superior National Forest and the Boundary Waters, which is managed by the Forest Service. So question number one is, why is the Forest Service not one of your partners?
That list was not at all exhaustive. So there are many, many, many partners. And of course, we have to have permits to do this from the Forest Service. So they are definitely a signed on partner for all of the projects we have in Superior National Forest in its entirety, as well as within the mountain. Yeah, we work with the office in Duluth. Trent Wickham has been kind of our liaison, who's a scientist there, who works both on the Minnesota side and the Wisconsin side. Yeah. That was an admission only of there are too many partners.
Got it. Second, I'm curious about how your work and the Science Museum's work is translated into public policy. So a lot of your science can, and I would say should, inform policy decisions on land management, on water quality, regulations, so on and so forth. And so I'm just wondering if there is an advocacy component, if you will, of using your science and your research to inform decision-making at both the local, regional, state, and national level.
Yeah, absolutely. I'm glad you said informed policy. That's actually one of our missions at the Science Museum. There's been several concrete examples of that. So you may be aware that there are nutrient standards for all lake in Minnesota, and those standards are divided by ecoregion and by depth. That was actually based on a study done by the Science Museum of Minnesota, where we went and basically cored lakes all across the state, all the different ecoregions. And we used that paleolimnological method to look at what were these lakes like 150 years ago to set a baseline goal. So we can't, because monitoring in many cases only goes back, if we're lucky, 20 years, at which time many of these lakes were already impaired by most standards. We had to figure out a way to actually go back even further to see what could we possibly, what's the best we could attain, and then realistically, what can we attain now that we know that the world is never going to be like it was 200 years ago. So that science from Science Museum researchers was used as the baseline data that determined where they set those standards for the entire state of Minnesota. We have other examples. So the trickle sand ban, that was based on sediment core work done by scientists at the Science Museum, where we looked at trickle sand. So trickle sand is an antibacteria that was used to be in like every hand soap product that you could buy. And we looked at how it accumulated in the aquatic system using that same sediment core methodology. So we showed that this is not something that washes through. This is something that stays, for decades. So that was, again, Minnesota was the first state in the country to ban trickle. Now almost all of it. So that's another. We work very closely with agencies. All the studies I talked about today have either direct agency partnerships, so either the Pollution Control Agency, the Department of Natural Resources, County, Lake County, St. Louis County, Soil and Conservation Watershed Districts. So we work with those people who are working for the government at the landscape level to inform them of, to work with them on the science we're doing so they can put together some of the policy reports to go into legislation. But we also work directly with legislators. Science Museum has a legislative liaison who works with legislators and tells them about the things that we're finding, what we think are important, what we think need to be on their radar. We attend the water quality salons at the Capitol, where they have legislators who are interested in water quality issues. We tell them, they ask us what we think are the most important issues. We give them our opinions on it, and we tell them the data that we've collected that support that. So we're very active in that arena. It's a very important part of the mission as a museum.
Represent a Zoom question here, if you don't mind. Becky on Zoom, Becky Prang asked, could rusty crayfish also make more nutrients available to the cyanobacteria by consuming aquatic microphyte beds, which would otherwise take up nutrients from the water? Excuse me if I misrepresented the
Yeah,
word.
so rusty crayfish are a crustacean that is unfortunately spreading all over Minnesota. I don't study rusty crayfish, but anything that disturbs the sediment or competes with other primary producers or eats other primary producers, which could compete with cyanobacteria, could indirectly cause algae blooms to increase for sure. And that's something we're looking, that's kind of the complex piece of it. It would be like an hour talk in itself. There's all the different food web interactions. So things like zebra mussels. Well, at first we thought, oh, that's good. They're cleaning the water. But then we figured out, well, then they form these gigantic mats, which are anoxic at the middle. And that actually starts stripping the nutrients that they ate, they stripped out of the water. They actually then release again in a more labile form, so a more easily used form for algae. So now we're in the reverse. So you may have heard Lake Erie was clearing for a long time. Now that is reversed, because now that community has matured, we're seeing what actually happens. So yeah, there's all kinds of indirect food web things that happen. That's why aquatic invasive species are such an important thing to look at and to prevent.
Another Zoom question from Carl. We've been getting more windy days and stronger winds in recent years. Is there a known transfer of algae from one lake to another due to strong winds? Is it likely that the lake on Isle Royal was contaminated by algae being blown from Lake Superior water into Lake on the island?
So winds are an important factor that we're looking at, but not for the reason of translocation of algae. The lake Isle Royal, which had that algae bloom, is called Lake Ritchie. It's on the southwestern side of Isle Royal. The Superior itself that's around it is much cleaner than the inland lakes. I mean, it has much lower productivity. They're all very clean lakes. It's in the wilderness. So that's not the issue. The issue is the wind. You know, I mentioned earlier how we're interested in looking at those different layers, the top layer and the bottom layer. What we found is that we have increasingly strong storms, especially in the middle of the summer, when you think of like the doldrums, the dog days, that, you know, a storm that may blow 30-mile-per-hour sustained wind can actually mix some of those lakes that are only 20 or 30 feet deep prematurely. So usually those lakes would stay, those two layers in the lake would stay separated from spring until fall. And that whole time, there's all these nutrients building up in the bottom water, but they're never able to be used by any of the primary producers because it's dark and cold down there. But what happens is if you have more frequent windstorms, if you reduce the amount of protection of the lakes themselves by removing the forests surrounding it, you get a mixing event, which then all of a sudden releases all those nutrients all in one big event, like literally in an hour. So I mentioned we have these buoys out that are taking these readings every 30 minutes. We did a similar study in southern Minnesota where we had the same kind of setup. And we thought, you know, in one lake in particular, it was Madison Lake, in the town of Madison. It's about 30 feet deep. The lake, and most years we expect to be stratified for the entire year. That one summer, one of the summers we were out there, there was a storm that blew 60-mile-per-hour straight line winds. It mixed the whole lake in August, and there was just a gigantic algae bloom which followed that, and then basically continued throughout the entire rest of the season, because once you mix that cold water, you've lost it. You know, you're never going to get the water that cold again, because that water is like 35 degrees at the bottom of some of those deep lakes. Once it gets mixed with the water that's sitting at 70, it's not going to cool down again until fall. So now you just basically spin your lake around for the rest of the season.
This will be our last question, Mr. Alison Brown.
You mentioned how many trips you take your shore and so on. So much of the problem seems to stem from climate change. So what is the agency, the museum doing to switch over to electric vehicles? So you can reduce your carbon footprint.
So we actually, you want me to take this one, Allison?
Yeah.
Okay. I'll just say what I know, then Allison wants to elaborate any, and she can't, I'll let her do that. So I just had an email from the director of museum facilities last week where we're already starting to consider how we change all of our fleet over to EV vehicles, how we increase the accessibility of charging stations, which of course is the biggest limitation to, well, besides the cost, there's the cost, and then there's the charging station, the network. Those are the two biggest hurdles to going to EV. And for us, when we're spending 8 hours on forestry roads, there is not yet a vehicle which will allow us to do that work. We can charge up in Grand Marais, but we can't do a full day's work. But then we'll get there, and we are watching that, and we are ready to make that transformation as soon as possible. We're already starting to make it, for our urban.
Just expand on that in terms of our sustainability, environmental goals. Is we are striving to reach net zero by 2030. We have reduced our carbon footprint by 86%. And this through a whole variety of things. And I'm sure the group has looked at that last 14% is frequently the hardest percent to achieve. We have done, none of them have been easy. We've done the easiest things already, and so we're really looking at what are those last things that we can do. And there are different phases that you can do in carbon neutrality. And we're really striving to reach all of the different phases in our carbon neutrality. We have lots of things we need to look at. That you're going down the path of refrigerators and how many coffee makers we have and all sorts of things that are needed in that important conversation. Thank you very much.